What's up everybody? Today I'm here with a quick overview of 3 labs done in class on diffusion and osmosis. Let's get started. A cell's interaction with the environment are crucial. It is a give and take that never ceases. Without it, life could not exist. Living cells are encased within a lipid membrane through which a few water-soluble substances can pass. The membrane also contains protein passages that permit specific substances to move into and out of the cell, and allows the cell to exchange information with its environment. Many substances can move in and out of the cell without the cell having to expend energy. This type of movement is called passive transport. Diffusion and osmosis is primarily how substances pass through a membrane. Diffusion is the movement of molecules from an area of high concentration to an area of low concentration. In other words, a concentration gradient. Osmosis is the net diffusion of water across a membrane and follows its own concentration gradient from high to low water concentration areas. In this lab, the purpose is to determine and observe the process of diffusion and osmosis. Procedure. Step 1. The solutions were paired with each other differently and the cups were labeled. Step 2. The predictions of each pairing were recorded. Step 3. The chosen solution to work with was sucrose. Step 4. The dialysis tube was tied off of one end with dental floss and was labeled to the solution. Step 5. Approximately 10 milliliters of the solution was added to the graduated cylinder. Step 6. The solution in the graduated cylinder was poured into the dialysis tubing. Step 7. The dialysis tube was sealed off with dental floss and using an electronic balance, the mass was measured and recorded as the initial mass. Step 8. The dialysis tube was rinsed in tap water. Step 9. Steps 3 to 8 were repeated using starch, NaCl, and distilled water. Step 10. Each dialysis tube was placed in a cup according to its pairing. Step 11. The cup was filled with the correct solution pairing until the tube was submerged in the solution. Step 12. After 20 minutes, each tube was taken out, measured, and recorded as the final mass using an electronic balance. Observations were recorded throughout the lab. Pairing 1, it was predicted that water diffused out of the bag. Pairing 2, it was predicted that water diffused into the bag. Pairing 3, it was predicted that water diffused into the bag. And pairing 4, it was predicted that there was no net movement of water. Observations before and after the experiment were made. For all pairings, the bag contained bubbles. After the experiment, solutions were stuck on all bags, and the shape of the bags were: for pairing 1, it was deflated; for pairing 2, it was inflated; for pairing 3, it was inflated. For the control pairing, the shape remained the same. Factors that affect the rate and direction of osmosis and diffusion include solute and water to follow its own concentration gradient from high to low concentration. Temperature: higher the temperature means faster moving molecules across the membrane. Surface area. The larger the surface area means high rate of diffusion and osmosis. This is the reason why cells are small. In this diagram, the cubes represent the cells. On the left side, we have a large cell, and on the right side, we have many smaller cells. However, for the same amount of volume, you have a greater total sum of surface area for the many smaller cells. And this is beneficial to the cell because of the high rate of diffusion and osmosis in transporting nutrients and wastes. Pressure includes osmotic pressure. It is the pressure required to stop osmotic flow. Water solute potential. The direction of osmosis is from high to low water potential. Water moves towards the high solute concentration. Also, there is more water molecules that surround the high solute concentration, which results in less free water molecules that can diffuse across the membrane. And molecule size. Large molecules cannot pass through the membrane while small molecules can. The pairing of sucrose and NaCl. The prediction is correct. Water movement is from high to low water potential. Sucrose is greater than NaCl in solute potential, which means water will move from sucrose to NaCl. The result is a decrease in mass. Water moves towards the high solute concentration, and NaCl dissociate into Na ion and Cl ion. Pairing of NaCl and starch. The prediction is correct. Water movement is from high to low water potential. Starch is greater than NaCl in solute potential, which means water will move from starch to NaCl. The result is an increase in mass. Water moves towards the high solute concentration. NaCl dissociates into Na ion and Cl ion. The pairing of starch and sucrose. The prediction is wrong. Water movement is from high to low water potential. However, starch and sucrose equal each other in solute potential, which means there is no net movement of water. This results in an increase in mass. There is no net movement of water because the solute potential are the same. The result is due to errors in the lab. The pairing of distilled water and distilled water. The prediction is correct. Water potential of distilled water is 0. The concentration inside and outside the bag are the same, which means there is no net movement of water. The result is an increase in mass. There is no net movement of water because the solute potentials are the same. The result is due to errors in the lab. Sources of error. One possible source of error is when measuring and recording the mass of the dialysis bag. In the observation, the solutions were still on the bag. This contributes to the increase in the final mass. The second possible source of error is leakage. Not tying the bag tight enough allows for leaks. An increasing mass when there should be no net movement can be due to solution leaking into the bag. The third possible source of error is when pouring the 10 milliliter solution from graduated cylinder to each bag. Without rinsing the graduated cylinder, the residue from the previous solution impacts the concentration of the next solution. This impacts the direction of movement of water into and out of the bag. Dialysis tubing and cell membrane. The tubing is made up of cellulose, while the cell membrane is made up of phospholipid bilayer. The tubing has limited-sized pores restricting any large molecules such as glucose, while membrane has protein characters that large molecules such as glucose can pass through. The tubing diffusion direction can only follow a concentration gradient from high to low, while membrane has active transports that can move substances against the gradient. The digestion of starch to glucose. Starch is a polysaccharide and its size is too large to diffuse into cells. In this lab, it shows that starch does not cross the semi-permeable membrane. Glucose is required for aerobic metabolism. Without glucose, you halt the ATP production, because at the beginning of the Krebs cycle, glucose is used to split into 2 pyruvate. Without ATP, animals have a difficult time surviving. The inability to break down carbohydrate is carbohydrate malabsorption. It is important for the IV solution to have salts in it because this solution is directed into the bloodstream. By keeping the salt concentration outside the cell approximately the same as the salt concentration inside the blood cell, it replicates an isotonic solution for the cell. If the IV solution is pure water, the flow of osmosis continues to flow into the cell without stopping. This is because this blood cell contains a higher solute concentration and the water diffuses towards the higher solute concentration. This represents a hypotonic solution for the cell. The blood cell is going to burst or is in lysis. A blood sample is necessary in measuring the solute concentration inside the blood cell. By experimenting with different concentration, the right amount of solution concentration is when the cells are in an isotonic solution. This allows for the matching of the same solute concentration inside the IV bag to that of the solute concentration of the blood cell.
Now let's look at the process of osmosis within a living cell. Plant cells and fungi have a cell wall that exerts a resistance to water movement, also known as the Tudor pressure. When the cell is in a hypertonic solution, the concentration of the water molecules in the cell is higher than the outside. Water molecules tend to flow from higher concentration to lower concentration. Therefore, the cell would experience a net loss of water. When this occurs within a plant cell, it is called the plasmolysis. On the other hand, when the cell is in a hypotonic solution, the concentration of water molecules in the cell is lower than the outside. Water would flow into the cell from outside so that the cell would experience a net gain of water. When the cell is in an isotonic solution, the water concentration inside and outside the cell is the same, which results water flowing in and out of the cell equally. Therefore, the purpose of this lab is to observe the osmosis process within a living plant cell. Procedure. Step 1. Peel off a piece of the epidermis of the red onion. Step 2. Place the onion on the glass slide. Drop a few drops of water on top of it and cover it with the cover slip flatly. Step 3. Locate a section of the onion skin under low, medium, and high power. Observe. Step 4. Add 2 to 3 drops of sodium chloride solution onto one edge of the cover slip. Drop the NaCl solution across the slide by touching a piece of paper towel to the foot under the opposite edge of the cover slip and observe through the microscope. Step 5. Remove the cover slip and flood the red onion cells with fresh water and then observe under high power. Step 6. Step 4 was repeated for aqueous solution. Observation. The plant cells are surrounded by distilled water. Every cell is filled with cytoplasm that is the rich purple color completely. The plant cell was surrounded by NaCl solution. The purple area within the cell is shrinking down slowly. When plant cell is surrounded by distilled water the second time, the lessened purple color area is now getting larger and filling the cell again. When the plant cell is surrounded by aqueous solution, the cytoplasm shrinks down again in a more obvious manner. Here's the demo for the complete observation for the lab. What will happen if we apply salt water to the roots of a plant? The salt water places the cell in a hypertonic solution, which means the water molecules inside the cell will flow out due to osmosis. However, because of the rigid structure of the cell wall, the cell as a whole is not able to shrink down, which means the water storage of the plant cell, that is the vacuum and cytoplasm, will shrink down, and this process is scientifically known as the plasmolysis. Water potential is defined to be the tendency for water to move. When the water potential inside the cell is higher, that means it has more energy for water to move. Therefore, the water molecules will move more frequently toward outside. What controls Tudor pressure? Tudor pressure is the pressure exerted on the cell wall by water passing inside the cell. Therefore, Tudor pressure is osmotic pressure. Due to the rigid cell wall, plant cells are not free to enlarge or shrink. Their Tudor pressure is controlled by osmosis and diffusion, which both are accomplished using a concentration gradient. When the cell is in a hypertonic solution, water flows out until it achieves an equal concentration both in and out of the cell. When the cell is in a hypotonic solution, water flows in to get the same result, which causes the cytoplasm in the vacuole to change shapes as the conditions apply. Contractile vacuoles are a type of vacuole that is involved in the osmoregulation process by helping pump out water from many water organisms. These cells live in a water-rich environment, which means water will flow into the cell consistently due to osmosis. This is why it's important for the contractile vacuole to act as a protective mechanism and expel the excess water out frequently, otherwise the cell would explode due to the rigidity of the cell wall. Contractile vacuoles are able to contract and squeeze out water periodically in order to make sure these one-celled organisms stay alive. For one-celled organisms that live in salt water, their surrounding environment will always be hypertonic. Therefore, the outer environment has a smaller concentration of water molecules compared to the inside of a cell, and water is more likely to flow out instead of into the cell. That's why there's no need for contracting vacuoles, since osmosis pushes water out perfectly. Bacteria and molds tend to grow in a warm and moist condition. In order to preserve food, people often put it in a hypertonic environment by adding salt or sugar. The water in the food will flow out due to osmosis, which leaves the food dehydrated and inhospitable for bacterial growth. Also, the removal of water from the food slows down its metabolism and hence helps keep it fresh longer. So, how do we find the water potential of plant cells in general? In order to determine the water potential of a plant, we need to determine the solid potential and pressure potential. The solid potential can be calculated using the formula psi as equals negative ICRT. I is related to the different types of the plant specifically, and R and T are known, which means we have to find the concentration of the solute. By graphing the percent change of mass of a certain type of plant cell over the known molarity of a certain solution that is differently concentrated, we can find the polar concentration of the plant cell, which is where the line of the graph crosses the x-axis. For example, we can use this method to find the water potential in the potato cells and carrot cells. After we let the same size potato cells sit in the sucrose solution with different concentration for the same amount of time, we can find the polar concentration of the potato cells approximately at a molarity of 0.3. Using this value, we can find the corresponding solute potential value. Since the pressure potential is very small in this case, it becomes negligible. Therefore, the water potential equals the solid potential, which turned out to be negative 860 kilopascal. The same procedure can be applied to carrots as well, and the value found for the solid concentration is approximately at a molarity of 0.8. The water potential is then calculated to be negative 2580 kilopascal.
In the diffusion osmosis challenge, 6 sucrose solutions with different molarities were given, along with the 6 possible molarities. To determine the molarity of each solution, the concepts of diffusion and osmosis were used. Pieces of raw potato were used to represent cells. The hypothesis for this lab was that the higher the molarity of the solution, the more water that will diffuse out of the potato cell. The materials used were a graduated cylinder, 6 plastic cups, a knife, a cylindrical piece of raw potato, 30 milliliters each of brown, green, red, blue, yellow, and purple sucrose solution, tweezers, a scale, a marker, a timer, and a petri dish. The potato was cut into 6 roughly equal-sized pieces. The mass of each potato was recorded. A piece of potato was dropped into a plastic cup. The plastic cup was labeled 1. 30 milliliters of the brown sucrose solution was measured in the graduated cylinder, then poured into the plastic cup. The potato was completely submerged in the solution. The time when the solution was poured in was recorded. The graduated cylinder was rinsed out. Steps 4 and 7 were repeated for the green, red, blue, yellow, and purple solution. The plastic cups were labeled 2, 3, 4, and so on. After 10 minutes of soaking in the solution, the potato was removed and placed in the petri dish. The potato's new mass was recorded. This table shows the mass of the potato pieces over the course of the experiment. This table shows the difference between the initial and final masses of each potato disc, as well as the percent change, which is given by the formula final mass minus initial mass over initial mass. We also observed that potato number 1, which is brown, and potato number 4, which is blue, sunk in the solution, while all the other potatoes floated. A solution with a high molarity would have more sucrose and less water compared to the other solution. In this case, the concentration of water inside the potato is greater than the concentration of water inside the solution. This would cause the water to diffuse from the inside of the potato to the outside potato, decreasing the mass of the potato. On the other hand, in a solution with a lower concentration of sucrose, there is less sucrose and more water. Thus, the concentration of water in the potato would be less than the concentration of water outside of it. Osmosis then causes the water outside the potato to diffuse down the concentration gradient into the cell, increasing the mass of the potato. Using that information, we can find that the green solution has a molarity of 1.0, while the blue solution has a molarity of 0.0. Using the percent change of mass as the y-axis and the molarity of the solution as the x-axis, we can graph the values of the green solution and the blue solution. By connecting these 2 points, we form a line. By plotting the percent change values of the remaining solutions, we find that they are relatively close to that line, connecting the percent change values of the green and blue solution. Thus, we can surmise that the other molarities have a common difference as well. The other molarities would then be 0.2m, 0.4m, 0.6m, and 0.8m. In conclusion, the more the percent decrease of the mass, the higher the concentration of the solution. The more the percent increase of the mass, the lower the concentration of the solution. Thus, the conclusion is, green has a molarity of 1, red has a molarity of 0.8, yellow has a molarity of 0.6, purple has a molarity of 0.4, brown has a molarity of 0.2, and blue has a molarity of 0.0, which is distilled water. The reason the potato pieces that sat in the brown and blue solution sank while the others floated is because in the brown and blue solutions, the solution was far less dense than the potato piece. The sucrose molecule is much larger than the water molecule. Thus, the lower the molarity of the solution, the less dense the solution. This matches up with the fact that the brown and blue solutions have the least molarity. Possible sources of error as to why the correct molarities of the solutions were not obtained are: number 1, the size of each potato disc was not the same. Because the larger potato discs have a larger surface area, water diffuses in or out of them more easily than in smaller potato discs. Number 2, the mass of the potato pieces could not be taken right after they were removed from the solution, as the scale was not always unoccupied. Thus, they had time to stay in the air and dehydrate for a period of time, decreasing the final mass of the piece. 3. The scale was a little wet from other groups using it at times. Thus, the residual solution left on the scale may have added to the final mass of the potato pieces. This graph shows the percent change in the mass of the potato core. It shows the relationship between the percent change in mass of the potato disc and the molarity of sucrose solution. The line of best fit, shown here in green, crosses the x-axis at the point 0.17. Thus, the polar concentration of the potato disc has a molarity of 0.17. If a potato core is allowed to dehydrate by sitting in the open air, would the water potential of potato cells decrease or increase? Why? In a flaccid potato plant cell, the pressure potential is positive and the solid potential is negative. Because the pressure potential and solid potential cancel each other out, the water potential is 0. However, when a cell is shriveled, pressure potential is 0 and the solid potential is negative as it does not change. Thus, the water potential is negative. It decreases. If a plant cell has a lower water potential than its surrounding environment, and if pressure is equal to 0, is the cell hypertonic or hypotonic to its environment? Will the cell gain or lose water? Explain. Because the water potential of the cell is lower than that of its environment, the solute concentration of the cell is higher than that of the environment. Thus, the cell is hypertonic to its surrounding environment in terms of solute concentration and gains water. If the water potential for a sucrose solution in a dialysis bag is negative 6.25 bars and is immersed in a cup of sucrose solution having a water potential of negative 3.25 bars, and if the water potential inside and outside the bag is 0, will the bag gain or lose mass? Explain your answer. Because the water potential of the sucrose solution inside the bag is lower than that of the sucrose solution outside the bag, the water will move from the outside to the inside of the bag. The osmotic potential of a solution with molarity 0.15 is negative 410 kilopascals, while that of a solution of molarity 0.20 is negative 540 kilopascals. Thus, the osmotic potential of solution of molarity 0.17 would be about negative 480 kilopascals.